This presentation will provide advice for routine HPLC analysis. This time we will look at baseline fluctuations and problems in reproducibility. I will now talk about the causes and countermeasures for baseline fluctuation. Here we have the two main causes of baseline fluctuation. First, I will discuss the effect of fluctuation of room temperature. Considering the laboratory environment in which the HPLC is operated, baseline fluctuations tend to be significant when the employed detector is sensitive against temperature variation and is installed near an air conditioner or near a door where people frequently enter and leave the room. In such cases, it may be necessary to take drastic measures such as changing the location of the HPLC itself. We will look at specific examples on the next slide. This figure shows the baseline drifting of a refractive index detector caused by temperature fluctuation. When the room temperature varies from 25 degrees Celsius to 35 degrees Celsius, the temperature of the THF mobile phase also varies, and baseline fluctuations can be observed even when a column oven is used. Please note that such drifting can also have a significant effect on peak integration, which affects quantitative calculations. This is an example of baseline fluctuation caused by dissolved air in the mobile phase. This figure shows the baseline variation in a reversed phase gradient mode using water and methanol as the mobile phase, monitored at UV 210 nanometers. The baseline drift caused by oxygen-derived UV absorption corresponds to an increase in the proportion of methanol, which has greater air solubility than water. However, when oxygen is removed by degassing the mobile phase, the degree of drift is improved significantly. Therefore, it is important to note that effective degassing of the mobile phase can prevent not only problems in mobile phase delivery but also baseline fluctuation. Next, I will talk about problems in reproducibility. Poor reproducibility of data in this case does not simply mean poor repeatability of peak area, but rather it refers to cases where the expected data cannot be obtained even with all the analytical conditions set according to the description in an official test method, published paper, in-house method, etc. Although all the advice discussed so far should be followed in this case, here we consider another important note related to mobile phase preparation and not other possible causes. If the mobile phase is not prepared properly, a variety of phenomena can occur including retention time fluctuation and peak shape deterioration. This slide explains the risk of preparing a similar but different mobile phase compared to the desired one depending on the description of the mobile phase composition. The description of mobile phase composition on the left should be interpreted that the mobile phase is a mixture of water and methanol, and that the mixing method is to take the two solvents in equivalent volumes then mix in an appropriate vessel. For example, if you mix 500 milliliters of water and 500 milliliters of methanol, the total volume after mixing will be slightly less than 1 liter, due to volume contraction, as you know. Therefore, exactly 500 milliliters of methanol is mixed in a mobile phase with a volume of less than 1 liter. This means that the methanol ratio is slightly larger than 50%, expressed as a percentage of volume. On the other hand, in the case on the right, the methanol concentration is specified as a percentage by volume, which means that exactly 500 milliliters of methanol is contained in 1 liter of mobile phase. Although the mobile phase compositions are similar, if you do not prepare the mobile phase with an accurate understanding of what is intended, you may encounter discordances in peak retention times, as shown in the next slide. This slide compares the chromatograms obtained by preparing the mobile phase with phosphate buffer and acetonitrile according to the notation on the previous slide. In the upper case, the mobile phase composition is defined by the volume ratio before mixing, and the mobile phase mixed with 900 milliliters of buffer and 100 milliliters of acetonitrile contains 100 milliliters of acetonitrile in a total volume of slightly less than 1 liter. The mobile phase in the lower case contains 100 milliliters of acetonitrile in an exact volume of 1 liter. Therefore, the upper case has a slightly higher ratio of acetonitrile in the mobile phase, resulting in faster elution of the compounds in reversed phase mode. Since the organic solvent ratio in reversed phase mode has a significant effect on retention time, it is important to prepare the mobile phase correctly as specified. A buffer solution has the effect of maintaining a constant pH and are usually prepared with a weak acid and its salt or base, or a weak base and its salt or acid. Generally, the concentration of a buffer solution should be described as the concentration of the weak acid or base that provides the buffering capacity, but if the description is ambiguous, the prepared buffer solution may have very different properties. Here 3 different interpretations of 20 millimoles per liter sodium phosphate buffer are compared. In the first example, 20 millimoles per liter is interpreted as the phosphate concentration and the paired sodium concentration is half of the phosphate concentration. By mixing an equivalent amount of undissociated phosphoric acid and fully dissociated monosodium phosphate, the pH would be the pKa1 of phosphoric acid. In general, this interpretation should be used for the buffer preparation. The following is a case in which 20 millimoles per liter is interpreted as the sodium concentration. In this case, a 20 millimoles solution of monosodium phosphate aqueous solution is prepared at first. The pH of the solution itself will be above 4. The pH is then adjusted to 2.5 by adding drops of phosphoric acid using a pH meter. In this case, the sodium concentration will not change, but the phosphate concentration will be greater than 20 millimoles per liter because of the addition of phosphoric acid. The next case is when the specified concentration of 20 millimoles per liter is considered same for both phosphate and sodium. In this case, some acid besides phosphoric acid has to be added into the monosodium phosphate solution to adjust the pH to 2.5. Since the phosphoric acid concentration is fixed, we use perchloric acid to adjust the pH to 2.5. All buffers can be considered 20 millimoles per liter sodium phosphate buffer, pH 2.5, but do they really have the same performance as a mobile phase? This slide shows the actual analyses using the 3 different mobile phases considered 20 millimoles per liter sodium phosphate buffer, pH 2.5, prepared by the 3 different preparation methods introduced in the previous slide. The analytes of interest were acetaminophen, dihydrocodeine, and caffeine, which are compounds commonly found in cold medicines. Of these, acetaminophen and caffeine are neutral compounds, while dihydrocodeine is a basic compound and positively charged in aqueous solution. The charged target compound is more polar, or in other words, less hydrophobic, than the uncharged neutral compounds, resulting in smaller retention in reversed phase mode. Furthermore, positively charged basic compounds interact with the negative charge derived from silanol on the surface of the silica gel packing material, causing undesirable adsorption phenomena such as a serious peak tailing. As a countermeasure, the mobile phase is generally adjusted acidic to suppress the dissociation of silanols, and at the same time, a negative charge is introduced into the mobile phase to form ion pairs with basic compounds to reduce the charge and suppress adsorption. From the above, the acidic mobile phase condition is a reasonable setting to inhibit the dissociation of silanols. Furthermore, by using a buffer solution instead of a simple aqueous phosphoric acid solution, it is thought that at a pH near the pKa1 of phosphoric acid, half of total phosphate molecules are dissociated having monovalent negative charge in the mobile phase. The dissociated monovalent phosphate ions form ion pairs with dihydrocodeine with a positive charge. In chromatogram A, the retention time of dihydrocodeine overlaps that of acetaminophen, but in chromatogram B, as explained above, the concentration of phosphate buffer is approximately double that of A, and the concentration of monovalent phosphate ions is also larger, resulting in a larger ion pair formation rate with dihydrocodeine. This is thought to provide longer retention, as the rate of paired meaning effectively uncharged dihydrocodeine also increases, and the hydrophobic retention in reversed phase mode becomes larger due to its lower polarity. Furthermore, in the case of chromatogram C in the lower panel, perchlorate ions used for pH adjustment can easily form ion pairs with positively charged compounds very efficiently. This is thought to provide a larger disappearance rate of the charged dihydrocodeine molecules than that in case of employing only phosphate buffer, resulting in increased hydrophobicity and longer retention. Therefore, Be careful with interpretation of analytical procedures, as you may encounter unexpected problems if the description related to analytical conditions is not correctly understood, such as the mobile phase preparation method. For your reference, this slide provides a detailed description of how to prepare an acidic phosphate buffer solution, without the use of a pH meter. First, the description for buffer solution. Buffer solution starts from the concentration as shown here, and the counter-ion species are listed in parentheses to indicate that the concentration is for phosphate only. This shows that the indicated concentration is only for the phosphate concentration, the counter-ion is sodium, and its concentration is separate from the phosphate concentration. Also, since the specified pH is 2.1, which corresponds to the pKa at a 100 millimoles per liter phosphate concentration, we know that the phosphoric acid and monovalent phosphate ion should be prepared in equivalent amounts, i.e., 50 millimoles per liter each in this case. In the case of 10 millimoles per liter, a 100 millimoles per liter buffer solution should be diluted tenfold or prepared according to the equivalent relationship shown in 2. However, since the pKa of a weak acid varies slightly depending on its concentration, the pH of the buffer solution will be 2.6 for the phosphate concentration of 10 millimoles per liter. Thus, when buffer solution is prepared using pKa, there is no need to use a pH meter and tedious calibration for each preparation. Only a balance and a measuring cylinder are required. We encourage everyone to put this preparation method into practice. As you can see from this and previous presentations, the reliability of HPLC analysis can be greatly improved by carefully analyzing the cause of an encountered problem and taking simple countermeasures. I hope that this lecture will be a help for you in your daily work using HPLC analysis. Thank you for your attention.